Hello everyone. In today's lecture, I will talk about the histology of the circulatory system. The learning competencies for this topic include: at the end of this lecture, the students should be able to identify elastic and muscular blood vessels, capillaries under the microscope. They should be able to describe the various types and structure-function correlation of the blood vessels. A student should also be able to describe the ultrastructure of the blood vessels. The circulatory system is a transport system for the movement of all body fluids. The principal functions include transport of oxygen and nutrients to the tissues, transport of carbon dioxide and metabolic waste products from the tissues, temperature regulation, and distribution of molecules such as hormones and cells like those of the immune system. The circulatory system has two functional components: the blood vascular system and the lymph vascular system. In this lecture, we will be discussing about the blood vascular system. The blood circulatory system comprises of a heart, which is the central muscular pump that initiates the blood flow. Then there are the arterial system, which comprises of the arteries and the arterioles. These distribute the blood further to the capillaries and the post-capillary venules. The capillaries and post-capillary venules are the actual sites of exchange, and the circulation through these vessels is known as microcirculation. From here, the blood goes into the venules, the small veins, the medium and the large veins, and finally back to the heart. We are going to discuss about all these vessels in this lecture. Apart from these vessels, there is also a lymph vascular system that you see here in green color. The function of this system is to return the excess extravascular fluid, that is the lymph, to the blood circulatory system and to transport the lymph to the lymph nodes for immunological screening. The whole of the circulatory system has a common basic structure. The three basic layers from inside to outside are the tunica intima, tunica media, and the outermost tunica adventitia. The tunica intima further comprises of three layers: the endothelium, subendothelial connective tissue, and internal elastic lamina. Modifications of this basic design accounts for various types of blood vessels, and as we go further, we will discuss how in different blood vessels there is variation in these three layers. But first, I will describe the basic structure of each of these layers in general. So let's discuss about the innermost layer of any blood vessel, that is the tunica intima. This layer, as you can see here, this is a transverse section of a blood vessel wall. The lumen is on this side. So the innermost layer here, this is the tunica intima. It comprises of an endothelium, which is the lining epithelium of this blood vessel. It is made up of simple squamous cells. So these are flattened in the transverse section These cells are basically polygonal, and the long axis of these cells is directed along the direction of the blood flow. These cells have a flat nuclei and scant amount of cytoplasm, and you can see that where the nucleus is located, there is a slight bulge in the cell. Now, these cells are supported by an underlying basement membrane. So all these cells are resting upon a basement membrane. This basement membrane is secreted by the endothelial cells itself. The endothelium contributes to the structural and functional integrity. It also maintains a selectively permeable barrier. It also releases substances that produce vasodilation and relax the blood vessel. These substances include endothelium-derived relaxing factor and nitric oxide. Outside this endothelium is a thin layer which is called the subendothelial connective tissue layer. This connective tissue layer is made up of loose connective tissue. Outside this is the internal elastic lamina. This layer separates the tunica intima from the tunica media. The internal elastic lamina is basically made up of fenestrated elastic material. These fenestrations help in diffusion of nutrients into the cells that are deeply seated in the wall of the blood vessel. You know that the innermost part of the blood vessel wall receives nutrition directly from diffusion from the blood. So this diffusion has to go into the deep-seated cells, and that is why the fenestrations in the internal elastic lamina help in this. Coming to the tunica media. The tunica media is a layer made up of smooth muscle fibers, collagen fibers, elastic fibers, and reticular fibers. The smooth muscle fibers in this layer are concentrically arranged, and you know the structure of a smooth muscle cell. It is a spindle-shaped cell with an elongated single nucleus in the center. So there are smooth muscle cells in the tunica media. Interspersed in between the smooth muscle cells are the elastin fibers, collagen fibers, and some reticular fibers. These fibers and the extracellular matrix is actually produced by the smooth muscle cells in the tunica media. There are no fibroblasts here to produce the various connective tissue fibers. This layer, the tunica media, is relatively thicker in the arteries compared to the veins. And in this layer, the relative amount of the connective tissue fibers and the smooth muscles depends on the type of blood vessel. As you go ahead, you will see that different blood vessels have different relative amount of smooth muscle and connective tissue fiber in this layer. Then, an external elastic lamina separates the tunica media from the outermost layer, that is the tunica adventitia. The tunica adventitia, it is the outermost layer made up of connective tissue, and in this layer the collagen fibers are longitudinally arranged along with some amount of elastic fibers. This layer gradually merges with the surrounding tissue, and it serves to anchor the blood vessel to the neighboring structure. This layer also contains the vasa vasorum and nervi vasorum of the blood vessel. This layer is relatively thicker in veins compared to the arteries. Now, let's talk about the structure of the arteries. Arteries are those blood vessels that carry blood from heart to various tissues of the body, and they mostly carry oxygenated blood except for the pulmonary trunk and pulmonary artery in the adults and the umbilical artery in the fetal life. Now, arteries are thicker than the veins. The arteries divide repeatedly. So initially, the arteries coming out from the heart are the large or elastic arteries, followed by the medium or muscular arteries, and the smallest arteries are the arterioles that open into the capillaries. Coming to the structure of the largest arteries, that is the elastic arteries. Elastic arteries are also called conducting arteries because they serve the purpose of conducting blood from the heart to the other smaller arteries. These arteries include the aorta and the pulmonary artery, and the large branches of aorta, that is the common carotid artery, brachiocephalic artery, subclavian artery, and the common iliac arteries. Now these arteries are called elastic arteries because they have a lot of elastic fiber in the tunica media. These arteries are capable of stretching to receive the blood from the heart during systole, and during diastole they are capable of elastic recoil that helps to come back to their initial lumen size. Thereby, these arteries facilitate a continuous and smooth movement of blood. What are the layers in the wall of the elastic artery? First of all, the innermost layer, that is the tunica intima. This layer is made up of endothelial cells, which is basically the simple squamous cells resting upon the basement membrane, and the long axis of the cells will be parallel to the direction of blood flow. The cells are joined by tight junctions. Underneath the endothelium will be a thin layer of subendothelial connective tissue, which is again loose connective tissue with some amount of collagen and elastin fibers. Now in the elastic arteries, some smooth muscle cells are present in the subendothelial connective tissue. These cells are contractile and they secrete the collagen and elastic fibers of this layer and also the extracellular ground substance. Outside the subendothelial connective tissue is a layer of the internal elastic lamina. It is a wavy layer of elastic fibers, but this internal elastic lamina is not very prominently seen in the elastic arteries. This is because the layer outside the internal elastic lamina, that is the tunica media, is itself rich in a lot of elastic fibers. So the elastic fibers in the layer of internal elastic lamina, they kind of merge with the tunica media and they are not very prominently visible. Now the tunica media of an elastic artery. This is a transverse section through the wall of elastic artery that has been stained with a special stain to highlight the elastic fibers. The previous figure was hematoxylin and eosin stain. This is a special stain, and you can see that in the tunica media there are large amounts of elastic fibers which are arranged concentrically. Also, there are some collagen fibers and some smooth muscle fibers. Now, This layer is the thickest layer of the elastic artery. As you can see, the span of the tunica media. The number of layers of elastic fiber varies with age. In the young age, the layers are lesser in number, and in the adults, there may be as many as around 50 layers to 70 layers of elastic fiber arranged concentrically in the tunica media. The smooth muscle cells present in this layer actually secrete all these elastic fibers and the collagen fibers. These elastic fibers are basically responsible for the elastic recoil of this elastic artery. These smooth muscle cells in this tunica media are actually said to be arranged in a low-pitched spiral relative to the long axis of the blood vessel. But in a transverse section, because of the low-pitched spiral, it appears to be arranged in a concentric fashion. The smooth muscle cells, apart from secreting the collagen, elastin, and the extracellular matrix, they are also important in repair of the blood vessel wall. Coming to the outermost layer of the elastic artery, that is the tunica adventitia. This is relatively thin compared to the tunica media. In the tunica adventitia, there is connective tissue with collagen fibers arranged longitudinally. These collagen fibers are type 1 collagen fibers, and they are responsible for the tensile strength of the elastic arteries. They prevent the expansion of the arterial wall beyond the physical limit during the systole. I told you that elastic arteries, they expand to receive the blood pumped by the heart during systole. This expansion is limited by these longitudinally arranged collagen fibers in the tunica adventitia, thereby imparting the tensile strength to this blood vessel. Apart from the collagen fibers, there are also fibroblasts, macrophages, and some adipose cells in this layer, along with the vasa vasorum that you can see here, the venules and the arterioles that supply nutrition to the vessel wall, and also the nervi vasorum, that is the nerves of this blood vessel. The nerves are basically sympathetic nerve fibers that secrete norepinephrine and therefore are vasoconstrictor in nature. Coming to the medium-sized arteries or the muscular arteries, they are also called the distributing arteries. The tunica intima of a muscular artery is thinner than in the elastic artery. It contributes to 1/6 of the thickness of the vessel wall in the adults. The tunica intima is made up of an endothelium, that is again simple squamous cells resting upon the basement membrane. Outside it is a thin layer of subendothelial connective tissue. This layer is very sparse in the muscular arteries and is made up of loose connective tissue with some collagen fibers. Outside this layer is the internal elastic lamina that you see here, this wavy layer. Now this layer is very prominent in the muscular artery compared to the elastic arteries. This is because in the tunica media of the muscular artery, there is more of smooth muscle compared to the elastic fibers. So the elastic fibers of the internal elastic lamina become very prominently visible. The tunica media of the muscular artery predominantly contains smooth muscle fibers. So the spindle-shaped smooth muscle cells, they are concentrically arranged. Roughly about 30 layers of smooth muscle fibers are seen in the muscular arteries. Apart from this, small amount of elastic fibers and some collagen fibers are also visible. The tunica media is separated from the tunica adventitia by the external elastic lamina present outside this layer. Now, the outermost layer, that is the tunica adventitia, its thickness is almost similar to that of the tunica media in case of muscular arteries. If you remember, in the elastic artery, the tunica media was much thicker than the tunica adventitia. Here, the relative thickness of the two layers is similar. This layer also contains longitudinally arranged type 1 collagen fibers along with some fibroblasts, elastic fibers, adipose tissue, and also the vasa vasorum and the nerves. Contraction of the smooth muscle of the tunica media of the muscular artery, it regulates the amount of blood flowing into the particular region. That is why the muscular arteries are called distributing arteries. The next type of blood vessel is the arteriole, also called resistance vessels. When the muscular arteries are traced distally and the diameter becomes less than 100 micrometer, then these are called arterioles. The arterioles have a very thin wall compared to that of muscular artery and the elastic arteries. You can appreciate this fact in this picture. Here you can see the wall of an arteriole and you can compare this to the much larger and much thicker muscular artery. Now the arterioles are further of two types, muscular arterioles and terminal arterioles. Muscular arterioles are the larger arterioles with diameter of 50 to 100 micrometers. The internal elastic lamina may be absent in a muscular arteriole and there are very few layers of smooth muscle in the tunica media. The terminal arteriole is much smaller. The diameter is less than 50 micrometers and there is only 1 to 2 layer of smooth muscle in the tunica media. The terminal arterioles, they give off lateral branches which are called meta-arterioles. In the meta-arterioles, there is slight thickening of the smooth muscle in the initial segment. This thickening is called the pre-capillary sphincters. These pre-capillary sphincters can shut off the blood flow into the capillary bed. So they regulate the blood flow into the capillary bed, and depending upon the metabolic activity of the particular tissue at that time, the precapillary sphincter will be open or closed. So if the tissue is metabolically very active, at that time the precapillary sphincters will be open and blood will be flowing through the capillary bed. But when the tissue is inactive or metabolically inactive, at that time the precapillary sphincters will be contracted to prevent blood flow into the capillary bed, and the blood will flow directly from the arterioles into the post-capillary venules. Let's now discuss about the capillaries. They are the smallest blood vessels. The capillary network is very large and elaborate. Thereby, it provides a large surface area and the blood flow is very sluggish in the capillaries. Both these two factors, the large surface area and the slower blood flow, they facilitate actual exchange of various substances across the capillary wall into the tissue fluid. The average diameter of the capillaries is 8 micrometers and it is just sufficient enough to allow the passage of RBCs one at a time. The capillaries, the density of the capillary network depends on the metabolic activity in the tissue. So the tissues with high metabolic activity will have a very dense capillary network. And the function of capillaries is exchange of nutrients and waste products between the blood and tissue fluid. The structure of capillary is basically endothelium resting upon the basement membrane. So there is a single layer of endothelial cells and sometimes outside the basement membrane there are specialized contractile cells which are called pericytes. So the pericytes and the endothelial cells, they share a same basement membrane. These pericytes, they support and control the size of the lumen of the capillaries. There are three types of capillaries: continuous, fenestrated, and discontinuous capillaries. Continuous capillaries are those where the cells in the endothelium are held together by tight junctions. So you can see here there is a tight junction between the two adjacent endothelial cells. So the transport of the substances will happen across the endothelial cell and it takes place by pinocytosis. And you can see a number of pinocytotic vesicles in the cytoplasm of this endothelial cell. Such capillaries are the most common in the human body and they are located in the skin, muscles, lungs, and brain. The fenestrated capillaries have got wide pores in between the endothelial cells. These pores or fenestrations are usually closed by a diaphragm which is made up of mucoprotein. So you can see the pores here in this capillary wall. Such fenestrated capillaries, they are present in endocrine glands, pancreas, intestinal villi, renal glomeruli, and the choroid plexus. Now the discontinuous capillaries, also called sinusoids. The sinusoids are large, irregular vascular spaces. The wall is very thin and you can see that there are large pores or slits between the adjacent endothelial cells. These endothelial cells rest upon a basement membrane which is also discontinuous. There is a very sluggish blood flow through the sinusoids and this makes for the good exchange between blood and the tissue fluid. Sinusoids are prominently seen in the spleen, liver, and the bone marrow. Sometimes the lining of the sinusoids may contain special phagocytotic cells, for example, the Kupffer cells seen in the liver. Other specialized cells may also be present in the sinusoids. Example, the cells of Ito, again found in relation to the sinusoids of the liver. So just to recapitulate, this is a continuous capillary with tight junctions between the cells. This is a fenestrated capillary where there are small pores or fenestrations which are closed by diaphragm. And this is a discontinuous capillary or the sinusoid where there are large pores between the endothelial cells and the basement membrane is also discontinuous. Coming to the venous system, the veins have much thinner walls compared to the corresponding arteries. However, the diameter is larger compared to the corresponding artery and the lumen is also larger. The blood flow in the veins is at a very low pressure. Many of the veins have valves in them to maintain a unidirectional blood flow. Exceptions, that is the veins that do not have valves, include the veins in the central nervous system, the inferior vena cava and the superior vena cava. On the basis of size, the veins have been classified as venules, small vein, medium vein and large vein. Let's discuss them in detail. Venules are basically the post-capillary blood vessels that collect blood from the capillaries. Their basic structure is that they have an endothelium with a basement membrane, as you can see here, and there is a thin adventitia which contains longitudinally arranged collagen fibers. Now, the smaller venules also have a specialized cell called pericyte outside the basement membrane, whereas the larger venules or the muscular venules have got smooth muscle. As you can see in this picture, this is a muscular venule. In the muscular venules, there is smooth muscle present outside the basement membrane of the endothelium. Now there are some specialized venules. These are called the high endothelial venules. These specialized post-capillary venules are actually lined by cuboidal or columnar endothelial cells resting upon a prominent basement membrane. Outside the endothelium, a prominent pericyte sheath is also there. These high endothelial venules are seen in secondary lymphoid tissues such as the lymph node. These venules have a special property, that is, they have a considerable permeability and they allow the passage of lymphocytes into the lymph node. Let's now discuss about the medium-sized veins. Again, in the medium-sized vein, the diameter is roughly 1 to 10 millimeters. Examples include the deep veins that accompany the arteries. These veins are characterized by valves, especially in the lower limb. Now, the tunica intima of the medium-sized vein is very thin and it comprises of endothelium with a basement membrane followed by subendothelial connective tissue layer which is also very thin and a discontinuous internal elastic lamina. Outside that is the tunica media which contains very few smooth muscle cells. The smooth muscle cells are arranged in circular fashion. The outermost layer is the tunica adventitia, which is thicker than the tunica media and contains collagen fibers and some elastic fibers. Large veins are those which have a diameter of more than 10 millimeters. In the large vein, as you can see here in this transverse section of a large vein, again there is a tunica intima, that is the innermost layer, which is further made up of endothelium resting upon a basement membrane, and the endothelium is simple squamous cells. Underneath it is the thin layer of subendothelial connective tissue and a poorly developed internal elastic lamina. Outside this is a layer of tunica media, which is also relatively thin and contains circularly arranged smooth muscle cells. Now, the outermost layer is the thickest in case of large veins, and this tunica adventitia in case of large veins contains longitudinally arranged bundles of smooth muscle cells. Since the smooth muscle fibers are longitudinally arranged, so in a transverse section, you will get this kind of a view of the smooth muscle cell bundles. Apart from this, in the tunica adventitia, there will also be the presence of the vasa vasorum, the arterioles and venules, and the collagen fibers, elastic fibers, and fibroblasts. Let us now discuss some clinical correlates associated with the histology of these blood vessels. The first condition is atherosclerosis. In this condition, the basic lesion is an injury to the epithelium. You can see here that in the top view, the endothelial cells appear polygonal, and these are the layers outside the endothelium. Now, the endothelium undergoes an injury because of several factors like hypercholesterolemia, hypertension, smoking, infection due to certain bacteria or viruses, and also advanced age. Then what happens is the macrophages will arrive here. You can see here the monocytes, they are migrating into the tunica intima and they become macrophages over here. And when they arrive here, they deposit lipids and some fibrofatty plaques are formed within this wall of the blood vessel. Now, the capsule of this fibrofatty plaque is formed by some smooth muscle cells and collagen fibers. Now, when this fibrofatty plaque ruptures, platelets will arrive here and they attach here, leading to formation of a clot or a thrombus, and all this finally leads to occlusion of the blood vessel. So this is a very simplified version of the sequence of events that happens leading to formation of an atheromatous plaque. In this picture, which is a transverse section of a blood vessel, you can appreciate that this is the internal elastic membrane and the rest of the layers outside it, they are so thin, and you can see the thickness of the atheromatous plaque within the tunica intima, and you can appreciate that the lumen has considerably narrowed in this blood vessel. So this is atherosclerosis. Then there is hypertension, which is basically high blood pressure. It is caused by reduced lumen of the small muscular arteries. This could be because of more smooth muscle within the tunica media leading to a reduced lumen, or due to contraction of the normal amount of smooth muscle that was present in the wall of the blood vessel. Another interesting fact is about the coronary artery bypass graft. Usually some suitable arteries are accessed to provide the bypass graft, but sometimes the great saphenous vein of the lower limb, which is a long subcutaneous vein, is used in coronary artery bypass graft. Why this vein is used? Because unlike the other veins in the body, this particular vein, the great saphenous vein, contains a lot of smooth muscle bundles in the tunica intima, media as well as adventitia. Because of this, it is harvested for the coronary artery bypass grafts when arterial grafts are unavailable or when multiple grafts are required. So now let us summarize what we have learnt in this chapter. So we know that there are elastic arteries, muscular arteries, arterioles which are further muscular or terminal arterioles, and then the capillaries. So the differences, the basic differences, as we go from elastic artery to the capillary, the diameter considerably narrows or reduces. Now the basic layer in the elastic artery in the tunica intima include the endothelium, the subendothelial connective tissue, and the internal elastic lamina, but the internal elastic lamina is not very prominent here. In the muscular artery, all these three layers are there in the tunica intima, and the internal elastic lamina is very prominent. Muscular arteriole has again the endothelium, subendothelial connective tissue, and the elastic lamina, but elastic lamina may be present or may be absent. Terminal arteriole will not have the internal elastic lamina, and capillary is made up only of the endothelium. As we come to the tunica media, in elastic artery, there are more of elastic fibers, as many as 50 to 60 layers, and some amount of smooth muscle, whereas in muscular artery, there is mainly smooth muscle in the tunica media and very few elastic fibers. It is because of the very few elastic fibers that the internal elastic lamina is prominently visible in this vessel. The muscular arterioles have few layers of smooth muscle, 8 to 10 layers, whereas the terminal arteriole has only 1 to 2 layers of smooth muscle in its wall, and tunica media is absent in capillaries. The tunica adventitia is thinner in the elastic artery compared to the tunica media. The tunica media is the thickest layer in the wall of the elastic artery. In the tunica adventitia, there will be some connective tissue and elastic fibers along with the vasa vasorum and the nerves. Similar structure is seen in the muscular artery. However, there will be more of connective tissue, less of elastic fibers here. In the muscular arteriole, the tunica adventitia is thin. In the terminal arteriole, it is very, very ill-defined, and this layer is altogether absent in case of the capillaries. The differences between the arteries and the veins. As far as histology is concerned, the arteries have a much thicker wall compared to a vein. All the three layers, that is the tunica intima, media and adventitia, they are well defined in the arteries. They are not as well defined in case of veins. The tunica media of an artery contains more of smooth muscle and elastic fibers. The veins have less of smooth muscle and elastic fibers and more of collagen in this layer. The lumen of the arteries is smaller than that of the corresponding vein, and you can appreciate this fact in this picture. These are the corresponding arteries and the veins, and you can see here the lumen of the vein is larger, and because the wall is thinner and lacks smooth muscle, because of that the wall is easily compressible and the lumen appears collapsed. whereas the lumen of the artery, though it is smaller than the corresponding vein, the lumen is much more rounded. Now, the tunica media of the artery is much thicker than the adventitia, whereas in case of veins, the tunica adventitia is much thicker compared to the tunica media. Veins, most of the veins have got valves in them. Arteries do not have valves, and the flow of blood is at a very high pressure in the arteries compared to that of veins. Thank you.